Dr. K. Anandi. I am working as assistant professor in Department of Pulses, TNA, Coimbatore. Today I am going to lecture on self-incompatibility. Self-incompatibility means the fertilization of the same pollen grain with the same plant in the ovule of the same plant is not possible. It is incompatible. The selfing is not possible with such kind of genotypes. So self-incompatibility encourages cross-pollination. More than 300 species belonging to 20 different families of angiosperms, that is seed-producing plants, are having self-incompatibility. The definition for self-incompatibility is, in self-incompatible plants, the flowers will produce functional or viable pollen grains which will fail to fertilize the same flower or any other flower of the same plant or even of the same genotype. So, self-incompatibility discourages the self-pollination and encourages the cross-pollination. Again, it is different from male sterility in that here the pollen grain produced is functional. It can fertilize the cross-compatible plants, but not the self-incompatible plants. So the self-incompatibility reactions are of 4 different types. That is, the pollen failed to germinate at the stigmatic surface, and next is it will be failing to penetrate the stigmatic surface, but it will be germinating. Then even if penetrates, it will be very slow in growth, and in the stylar reaction, the flower will drop before it reaches the ovule. Then even if the fertilization takes place, the embryo degenerates at an early stage. So 4 different types are there. First is in stigmatic surface, germination will not be there. Second is also in stigmatic surface, where it will germinate but penetration will not be there. Third will be in the stylar region, and fourth will be in the ovule. Even after fertilization, the ovule will be degenerating very early without production of seeds. The classification of self-incompatibility is given by Lewis 1954, that is heteromorphic system and homomorphic system. In heteromorphic system, 2 different types of style is reported, that is distyly and tristyly, where the length of the style will be different in the self-compatible class. Distyly, short style with long stamen and long style with short stamen is there. So the long style will be compatible with the long stamen, that is with the long stamen, the anthers produced in the long stamen, and the short style will be compatible with the short stamen produced from the short stamen which is in the another plant. Tristyly, here 3 different types of style will be there: short, medium and long. In homomorphic system, there will not be any difference in the flower morphology, that is homomorphic. It is of 2 different types again, gametophytic and sporophytic, where the gametophytic, the incompatibility reaction takes place with the gametes, that is the pollen and the ovule. Whereas in the sporophytic, it is the genotype of the mother plant that is responsible for the incompatibility reaction. So in heteromorphic systems, difference in the morphology of the flowers is reported to be of distyly and tristyly. The example for distyly is Primula species, where the pin and thumb flowers are reported. The pin flowers will be having long style like a pin and short stamens, and the thumb flowers will be having short style with long stamens. In tristyly, Lythrum is reported to have tristyly, where the style is of 3 different lengths: short, medium and long. In case of pin flowers, the style is long and the anthers are short. In case of thumb flowers, the style is short and the anthers are long. The long anthers can fertilize only the long style, and the short anthers can fertilize only the short style. The otherwise it is the incompatible reaction. For example, if the short anthers are fertilizing the same style, it will not reach the ovule. The pollen growth will be slow. So such kind of reaction will take place in pin and thumb flowers, and the compatible reaction will be only between pin and thumb flowers, or not between pin and pin or thumb and thumb flowers. The phenotype of the flower, that is the genotype of the plant, that is a sporophyte, is responsible for the incompatibility reaction. The genotype of the gametes if the genotype of the gametes are responsible for incompatibility reaction of the pollen, then it is gametophytic. And the incompatibility reaction takes place in the stylar region. For pin flowers, it is completely recessive genotype is said to be reported, and for thrum flowers, the genotype is heteromorphic. All S, if both S are crossed between together, then it will produce all S dominant type, but such plants will be incompatible in reaction and it will not produce any seeds. So, this pin flowers, that is completely recessive, will be compatible only with the hybrid, and between pin and pin it is incompatible mating, and between thrum and thrum it is again incompatible mating. If the pin is crossed with the thrum flowers, then 1:1 recessive and hybrid or the heteromorphic thrum will be produced. That is, if the small s is compatible is joining with the capital S, it will produce thrum flowers, and if the small s is joining with the small s, it will produce pin flowers. And in case of thrum into pin also, again the same, if the capital S is with the small s, it will produce thrum flowers, and the small s is small s, it will produce pin flowers in the segregation of 1:1. One thrum and one pin will be produced from a cross. This is the tri-style system reported in Lithium. The yellow arrow points out the style, that is the short style with long and medium anthers, and the medium style with the short and long anthers, and the long style with the short and medium anthers. So, the compatibility will be there only between the long style and the long anthers, and with the medium style and the medium anthers, and the short style and the short anthers. This short style will not be compatible with medium and long, which is produced by the same type of the flowers. Again, it will not be compatible with the long style produced by the other type of flowers, but it will be compatible with the short style, short anthers produced in the other flowers. So, here you can see that in case of short anthers, it will be compatible with the short style, and the long anthers will be compatible with the long style. And here 3 different types are there, where the short anthers will fertilize the short style, but the short anthers will not produce medium or long style. And again the medium anthers will not fertilize the long anthers, long style, but the medium anthers will be fertilized in the medium style. The same type morphology will be compatible, but the different type will be present in the same flower, but it will be incompatible. So, homomorphic system. Here the incompatibility is not associated with any morphological difference, and the incompatibility reaction is controlled by either the genotype of the parent or its own genotype. If it is the genotype of the parent, that is the spore producing parent sporophyte, such kind of reaction is called as sporophytic control, and if it is by the own genotype, then it is gametophytic control. That is, the gametes genes are responsible for the incompatibility reaction. The gametophytic system was first given by East and Mangelsdorff in Nicotiana Sandre during 1925. The incompatibility reaction of the pollen is determined by its own genotype and not by the genotype of the pollen producing plant. The incompatibility reaction is controlled by single gene having multiple alleles, and here with this codominance is assumed. The examples are Trifolium, Nicotiana, Lycopersicum, Solanum and Petunia. The incompatibility reaction goes like this. If it is S1 and S2, then S3 and S4 is compatible. If one pollen grain is S3 and the other one is S1, then S1 is incompatible, but S3 is compatible. If it is S1 and S2, then both are incompatible. Even if one gamete is different, because here it is gametophytic control and not the sporophytic control. And here codominance is reported. So, the compatible cross is either S1 into S1 S2 to S3 S4 or S1 S3 to S1 S2. Here it is partially compatible. Here it is completely incompatible, and here it is completely compatible. Here there is, this reaction is controlled by codominance. In sporophytic system, it was first reported by Hughes and Babcock during 1950 in Crepus fotida and by Gestel in Parthenium argentatum. It is controlled by a single gene. Again, here also multiple alleles are reported. More than 30 alleles are reported to be in Brassica oleracea. But here, codominance is not seen, but dominance is assumed. That is, if it is S1 is dominant over S2 and S2 is dominant over S3, like that it goes on. But in case of gametophytic system, there is codominance. All are equally dominant. It is reported in radish, brassica and spinach. Here the determination is by the genotype of the plant, pollen producing plant and the ovule producing plant, and the genotype of the pollen or ovule is not responsible for this reaction. Here again we can see the genotype of the plant is involved. Hence, if it is producing S1, S2, then it will behave like all S1. If it is S1, S3 also, it will be all S1, because S1 is dominant over S2 and S3 and S4. S2 is dominant over S3 and S4, and S3 is dominant over S4. So, the smaller number will be dominant and it will be reacting as a completely dominant one. Here if it is S1 into S2 and S1, S4 is there, then partial compatibility will not be there, but it will be either completely incompatible or it will be completely compatible. Here it is S1, S2 and here it is again dominant is S1. So, even if S2 is there, it will be compatible. So, here partial compatibility is not reported. If the two genes are different, then this S4 cannot germinate as because this S1 is here. Because these two will react together, as it is the spore producing parent is responsible, but not the pollen grain. Individual pollen grain is not responsible for the genotype. So, the characteristics of sporophytic system is given by Lewis. There is frequent differences between reciprocal crosses. So, if the female is taken as male and the male is taken as female, then it will become compatible. But such kind of reciprocal reaction is not observed in gametophytic system. Incompatibility reaction occurs only in the female parent. A family can consist of 3 incompatibility groups. Homozygotes are normal part of this system. An incompatibility group may contain 2 genotypes. Here homozygotes are produced, but in other system homozygotes is not produced. So, these are the differences between sporophytic and gametophytic self-incompatibility. The incompatibility reaction takes place in the sporophytic genotype in case of sporophytic, that is the spore producing parent. In the case of gametophytic incompatibility, the incompatibility reaction is determined by the genotype of the pollen, and it is mostly controlled by multiple alleles in both the cases, but in sporophytic system dominance is shown, and in gametophytic system it is without dominance, that is codominance is reported. The pollen tube is trinucleate in sporophytic and binucleate in gametophytic. The reaction occurs in the stigmatic surface in case of sporophytic, and in the gametophytic it occurs in the stylar region. Homozygotes are possible in case of sporophytic, but it is not possible in case of gametophytic. The incompatibility reaction is fixed in the pre-meiotic stage, that is the sporophyte of the parent is responsible. So, it should be before the meiosis. Once the meiosis takes place, it will become gametes. So, post-meiotic is reported in gametophytic self-incompatibility reaction. Mechanism of self-incompatibility. Pollen stigma reaction, pollen tube style reaction and pollen tube ovule interaction are reported. In case of stigma reaction, 2 different types of reaction we have already noticed, where the penetration will be stopped or the germination itself will be stopped. In case of style, the growth will be slow, and in case of ovule, even the fertilization takes place, the ovule will degenerate. So pollen stigma interaction is reported to be there in gametophytic and sporophytic system. Here the stigma is plumose and it is commonly wet stigma. So it will be there only in the stylar region. In case of sporophytic system, it is papillate and dry and it is covered by a hydrated layer of protein which is known as pellicle, which is involved in the incompatibility reaction. The pollen grain germinates and incompatibility reaction occurs at a later stage in case of gametophytic system. But in case of sporophytic system, the pollen grain releases an exine exudate which is protein or glycoprotein and it reacts with the pellicle and it induces callus formation and arrests the growth of the pollen type. So it will be controlled in the stigmatic surface itself. Pollen tube style interaction. Here the pollen tube germinates and the pollen tube penetrates the stigmatic surface and the incompatible combinations between the pollen tube and the stylar reaction takes place. That is, the growth is retarded within the style. It is reported in Petunia, Lycopersicum and Lilium. Protein or polysaccharide synthesis in pollen tube stops. Bursting of pollen tube takes place and the death of nuclei is what is there. In case of pollen tube ovule reaction, it is reported in Theobroma cocco, where the pollen tube reaches the ovule, but the fertilization also occurs, but it will the embryo will degenerate later due to some biochemical reaction. Then relevance in plant breeding. Hybrid seed production is produced by two self-incompatible but cross-compatible varieties. Alternatively, the seeds will be collected from both the lines as hybrids. So here, for example, if it is distyly, then the pin and thrum should be planted together. If both the type are two self-incompatible types, that is pin and thrum are planted, but they should be cross-compatible. If both are pin, then again it will become self-incompatible. So cross-compatible means pin and thrum should be planted. Planting of a self-incompatible variety along with another self-incompatible variety, then the seeds obtained will be self-incompatible. Example of this kind of self-incompatibility is reported in Brassica, Strover, Trifolium, Solanaceous, Astraceous crop. The difficulty is maintenance and production of inbred line. Hand pollination is also tedious and costly. Then the continuous selfing break down the self-incompatibility and self-fertile lines will appear. Such kind of things will be a problem in plant breeding as it will not produce hybrids as because it will produce self seeds also. So continuous selfing will lead to self-fertile plants, not the self-incompatible plants. So such kind of things will be then we will not be able to know whether it is because of crossing or it is a self disease. So environmental factors like height, temperature and humidity will also reduce the self-incompatibility. Then again, bees often prefers to stay with a particular parental line, which increases the proportion of self disease. So here hybrid production is also difficult and maintenance of inbred line selfing is also difficult in case of self-incompatible lines. Then elimination of self-incompatibility is done by various methods. That is to produce self-compatible lines. In case of single gene gametophytic self-incompatibility, doubling of chromosome will eliminate the self-incompatibility. Then mutation can also produce self-fertile lines. Then transfer of self-compatible alleles from related species through bracc crossing is also reported to break down the self-incompatibility. So these are the genetic methods, and here comes overcoming self-incompatibility by mechanical means, that is bud pollination, surgical technique, end of season pollination, use of high temperature, irradiation, grafting and double pollination. So bud pollination means application of matured pollen in immature stigma will break the self-incompatibility. Then surgical technique, here the stigmatic surface is removed or the stylar surface is removed based on where the self-incompatibility reaction takes place. If it is in the stigmatic surface, then the pollination will be done in the stylar region. If it is in the stylar region, then the style will be removed and directly the ovule will be pollinated. End of season pollination takes place it will favor the self-compatibility, but here the pollination at that time may be successful and may be unsuccessful also. Then use of high temperature, that is above 60 degrees, the exposure of pistil will induce pseudo-fertility. It is reported in trifolium. Then irradiation, X-rays and gamma rays temporarily suppress the self-incompatibility in solanacea. Then grafting. Grafting of a branch to another branch is reported to break self-incompatibility in trifolium pratans. Then double pollination, that is a mixture of incompatible lines and the compatible pollen grains, that is mentor pollen technique, is done in some. Then other techniques to overcome self-incompatibility are treating the flower with carbon monoxide, injecting the style with immunosuppressants, then electrical shock to pollen and stigma, that is 100 volts, then treating the stigma, that is a pistil, with phytohormones, then protein synthesis inhibitors can be used, then steel brush pollination can be used. In case of protein synthesis inhibitors, we saw that protein is pellicle is formed in the pollen and it will stop the pollen growth. So protein synthesis, if it is inhibited, then the pollen will start germinating. Again, steel brush pollination will disturb the stigma, dry stigma, and it will become wet stigma. Thank you.